with a similar similar genetic composition, then we're going to see that this cell has to multiply in size, and even the cell organelles have to increase in size in order to ensure that there's an equal number for each of the daughter cells respectively. Therefore, this particular stage, new cell organelles such as the ribosomes, the centrioles, the Golgi apparatus, and even the mitochondria are formed, whereby they increase in size in order to accommodate for the two daughter cells that I've said. And also there's, of course, genetic materials multiply in order to ensure that the same daughter cells are going to have the same number of organelles or the constituents as the parent cell. And basically we're going to see that the energy in form of adenosine triphosphate is going to have a build-up to ensure that the energy that is required in this process of cell division is given out or maybe produced. And then we're going to see that at this particular stage, of course, if we try to compare with this second stage for earlier prophase, the chromosomes are not basically observable or cannot be seen clearly at this specific point of interface. Now, therefore, we're going to see that this is going to proceed into the after the formation of a new organelles and the buildup of energy, which is ATP, and therefore we're going to see that it's going to proceed into this second stage, which is the prophase, earlier prophase. Therefore, at this specific stage, we're going to see that the centrioles which have been formed are going to separate and move to the opposite poles of the cell. This is the centriole that was here. We can see two pairs of sets here. They're going to separate and move into the opposite sides of the poles. Now, the pole in this case we refer to perhaps the left and the right side, or upper or lower. But in reference to this particular kind of illustration we're going to consider this left and right parts as our poles respectively. Now, in this case, we're going to see that this particular set of three processes, I'm going to treat this this first, earlier, middle, and the late prophase as a block. Therefore, when I'm going to do a discussion, I'm going to use either of these three illustrations in order to drive that point. So, as I've said, the centrioles will separate and move into the opposite direction of this specific part of the cell that we have referred to as poles. And we're going to see that the spindle fiber is going to start forming. So the spindle fiber is this specific thread-like structure that I'm illustrating on the board. So therefore, these thread-like structures are going to form unlike this earlier stage that I've given on the board. Now, this we're going to see that the formation of this circular envelope for the nucleus is going to start to disappear at this specific point. That is a prophase, this is the middle. And we're going to see that also the nucleus as a formation, the center is going to disappear, therefore result in this particular scenario that we're going to see here, where it's not basically shown as a circular part of that specific center. And in this case, of course, for the process of prophase, the chromatids and the chromosomes can be able to be seen clearly, and therefore this leads to another process that we refer to as